it's a real pleasure to introduce my colleague Greg Weiss here. So I've known Greg for his whole time here. I helped recruit him. Greg is a fantastic colleague. He comes from a very distinguished pedigree. He earned his bachelor's degree at that institution to the north with 25 endowed chairs, Paul Bartlett at Berkeley, where he worked on research in computational chemistry. He went on to do his PhD at Harvard and earned his PhD in 1997 with Stuart Schreiber in the broad area of chemical biology. He then took an unusual training turn, deliberately going into the private sector for the most expert training possible in the art of phage display. He went to our friends at Genentech, where he studied with Jim Wells and really learned to master this technique and learned to use it to probe biomolecular interactions, basically to tease out the roles of individual residues, individual amino acids in the interactions of proteins. He started his career in 2000 here at UCI. I was fortunate enough to be on the team that helped recruit him here and identify him as the very top faculty member on the market in chemical biology that year. He's cultivated a fantastic research program involving protein engineering, biosensing, diagnostics. He's developed a wonderful new test for prostate cancer that's going to have tremendous impact, and through all sorts of fantastic collaborations has moved in different directions, combining enzymology and electronics, and I think this is what we're going to hear about, some really wonderful work in mechanistic enzymology. So Greg really helped build the effort in chemical biology here. I would say before Greg's coming here in 2000, chemical biology was a little bit of a wasteland. In other words, using the tools of chemistry to probe the frontiers of biological science was very challenging because there wasn't this nucleus of expert faculty and expert students that in turn creates a whole network of expertise. And Greg launched a tremendous program of recruiting top students. He served as a focal point for recruiting faculty beyond himself, and now we have a thriving chemical biology community here, and it's really very much to Greg's credit. Greg has literally written the book on chemical biology along with Professor Dave Van Branken. He has written what I think is the preeminent textbook on the subject, and so students not only here at UCI but also elsewhere are having the privilege of learning from Greg and his training. Greg's interests and his impact go well beyond UCI. He thinks globally. He created the Global Young Academy, which is really fantastic, recognizing that in many other countries, young scientists aren't as fortunate as in the US. So in the US, we're really privileged here because an assistant professor comes into the department and is immediately able to launch a program of research independently from other faculty members, creating an environment that they can really exercise their intellect and create new ideas. And not all countries are this lucky. And so to bring together and help recognize the importance of young scientists, he created a network of young scientists, the Global Young Academy, that he served to lead until he aged out of it at age 40, or I think it was age 40. Anyway, on that note, I will turn the floor over to Greg. Let's give Greg a big warm welcome.
Thank you very much, Dave. Wow. What an introduction. What an amazing crowd. I'm taken aback. This is such a generous introduction, James. Thank you. And I'm also enormously honored to be giving this eminent lecture. I come from a department of truly eminent scholars and scientists, and this is a really big deal for me. So thank you very much. So today, I want to talk to you about the machines that power your cells. And I hope you got a chance to see the video as you were entering. That's to remind us that your cells are very busy places. There's a lot going on. There's sort of a constant movement of stuff and activity that's happening. And today I want to get into some of the details and tell you a little bit about this. This is a topic that's fascinated me ever since I was a little kid. I read this book by the great Isaac Asimov back when, well, I guess I must have been about 12 years old or something, and I absolutely loved it. This dream of being shrunk down to nanometer scale and then sailing around the blood vessels of a patient to try to cure medical challenges has always been my dream. It's always been my dream, actually, to be shrunken down to nanometer scale. Now, of course, we're not there yet, but I hope at the end of today, you'll know why this is going to remain science fiction, not just for the foreseeable future, but perhaps forever. So today, what I want to specifically talk about is, we'll give a quick overview, and I want to talk about this weird and wonderful nanometer scale universe, and what's happening when we zoom down to the tiny scale of single molecules. It turns out it's absolutely fascinating. And then we'll have a very brief introduction to what it is that the machines inside your cells do. And then I'll show you some new experiments that are taking place in my lab. And I'll show you 3 examples of the types of machines that my laboratory studies. These machines are called enzymes. They catalyze chemical reactions. And they're really remarkable in their abilities to do chemistry. We'll then take a closer look at lysozyme, because I want to share with you something that very few people on the planet know. It's a discovery that the laboratory just made that I'm really excited about. And we haven't even published it yet. It's still in peer review. It's going through the rigorous and long and protracted challenging process of peer review. But I want to share with you today some new results that I'm particularly excited about. And I think I hope you'll appreciate them as well. I also should tell you that I'm aiming for a general non-science audience. I'm taking this our charge for outreach very seriously. And for all of my distinguished colleagues, when I look out in the audience and I see you all, I really appreciate you coming out. Don't leave. But if you want more details, there are abundant details, and there is very detailed analysis and lots of data to back up what I'm showing you. But today, I'm going to keep things light, and I'll be up here, way up in the stratosphere and looking down on the big picture, and we won't be delving into all of that analysis and data. Okay? But if you want to see that, I'd be delighted to share it with you. We have lots of it. It backs everything that I'm telling you about. Okay, so let's get started. I want to talk to you, just start by reminding you what kind of scale we're talking about. This is an infinitesimally small scale range of lengths. We're going to be talking about objects that are down here in this nanometer scale. And of course, we humans are up here in the meter scale. And as you get down to smaller organisms, say ants, you're down in this millimeter scale. As you get down to individual cells, like red blood cells and the larger T cells, you're down in, say, the 5 to 20 micron scale. And then over here, we get to transistors. And then we get to individual molecules over here. So this nanometer scale is amazingly tiny. Everything is very, very small. This is a log scale. Okay, so just to give you, this is the compulsory comparison to the human hair. So if we had, say, a human hair running along the front of the stage, okay, so just imagine, not my hair, because I'm not giving any of them up, but someone else's hair running along this stage over here. The kind of little wires that I'm going to show you in a moment, if they were laid down in parallel to that human hair, and let's say my wires were a foot in diameter, then the human hair would start here, and it would keep going for 20 miles before you reach the other side of the hair, okay, in terms of its diameter, its width. Okay, so these are really, really tiny, tiny little things. Okay, and when we get down here, it turns out this is a very interesting universe. That there's all kinds of fascinating things that we see when we start looking at objects in this, with this great magnification. And I just remind you that magnification matters, right? As you start to magnify objects, our perspective on them changes. This is a beach, and the grains of sand look perfectly uniform. It looks like the perfect place to curl your toes. But if you were to zoom in on those grains of sand, you would find astonishing diversity. You'd find that each grain of sand is its own unique individual. It has its own shape, perhaps its own color, its own size. There's remarkable diversity. When we zoom down and start looking at individual molecules, we find a similar diversity that fascinates us, surprises us, and challenges us in really exciting ways. And that's what I want to talk to you about today. Okay, so it turns out this nano universe is a very weird and counterintuitive place. What we know in our macro world does not necessarily apply to the nanometer scale universe of individual molecules. So here we are in this nano universe. This would have to be someone not me, because I am strictly forbidden from writing wheeled objects. But over here in this macro universe, forces like gravity reign supreme, right? You can see this guy, fighting gravity as he's keeping his bike upright. Centripetal force is very important. Gravity is meaningless in the weightless world of individual proteins and molecules, and all of biology takes place in water. That's a weightless universe. Gravity, not an important force at all. It's important to us humans, not important at all to our molecules. Friction, very important to this guy. That's what's keeping him on the road. Not important at all in the biological universe. It's essentially frictionless. There's a different type of force that I'll tell you about on the next slide that's operative. Also, these objects are identical. This guy can go to the BMW dealership, pick up a new motorcycle, and expect it to have roughly the same horsepower, roughly the same characteristics. That's not at all what we see when we start looking at individual molecules. We find broad diversity. So this is what I mean when I say this is a weird world. Instead, what we find that there are other forces at work that are non-intuitive and extremely important. So this includes things like quantum effects. Quantum effects are important even in biology. They're spooky and strange and always surprising. I will not be talking about those today. I can have a whole separate lecture about those. Those are endlessly fascinating. But they are important. They do play roles in biology. Quantum effects don't really enter into our macro world. It turns out that molecules are constantly sticking to each other. Non-specific binding becomes a very important force. You throw a molecule against a surface, almost immediately it will stick down there. Clean surfaces do not exist in this universe. All the surfaces are coated with molecules and stuff. In addition to this non-specific binding, molecules also find each other in sort of hand-in-glove specific binding. Also very, very important. And then, as I alluded to on the previous slide, molecules are individuals. They have a certain graininess. They have diversity. They're not all the same. They're constantly different. They have different energies. Even molecules that should be identical, they're not identical. They have different energy levels or different vibrational nodes and things like that. We're going to see today that will factor into how the biology works. And then finally, there's a force that is unfamiliar to us, but it really is an important one, which is the thermal energy that's found in a glass of water at room temperature. That little tiny bit of room temperature energy has the water molecules vibrating around and banging into each other with tremendous forces. This is what drives Brownian motion. Okay, so Brownian motion is really a thermal storm. Molecules in water are being buffeted against tremendous, tremendous forces. If you were shrunk down to nanometer scale, like the fantastic voyage that I showed on the first slide, you'd be experiencing a hurricane that has wind forces of 15,000 miles per hour. Okay, that's obviously enough to tear apart anything that's not held together by covalent bonds, which is what molecules, that's how we define molecules. So it's amazing that the molecules are not ripped apart by these tremendously strong forces. Brownian motion was first described in 1909 by Jean Perrin, the French scientist. And actually, here's some of his data. Here he is tracking individual little particles in water. And you can see the particles are doing sort of a random walk. We could see this in this YouTube video as well. These are nanoparticles in water, and you can see they're jiggling around, they're getting buffeted by this thermal storm. This is really important. This is really what you experience if you're shrunk down to that nanometer scale. And as we're going to see today, your molecules have evolved to take advantage of this force. This force that doesn't mean anything to us up here in this macro scale. It's not something we think about, but it's going to be really important as we zoom down. Okay, so I've introduced you to the unusual nanometer scale universe. Let's talk a little bit about the cell's molecular machines. First thing is, your cells are a very crowded place. Even the exterior of the cells is very crowded. This is a surprise to me. Will you guys fall asleep if we go for no lights off? Okay. This wonderful picture is a watercolor by David Goodsell, and I'm a huge fan of his artwork, and I'll show a couple of images from him. And this illustrates a neuromuscular synapse. These are two cells talking to each other. The cell up here is a neuron, and it's releasing neurotransmitters into the joint between these two cells. This is called the synapse. And this is really how cells talk to each other, by releasing signals like carrier pigeons that then have to drift across this gap and reach the other cell to trigger some neuromuscular response. But notice that there's no voids here. There's no just water floating around. This whole thing is chock full of molecules that are depicted in these various colors. This is the way cells are. They are really jam-packed with molecules. It's like jelly inside there. The little tiny spaces that are apparently unoccupied in this image, those are filled with water. They're filled with salts. They're filled with other stuff. So there's really no vacuum space that's unavailable. These are very jam-packed areas. The other thing is that inside your cells, you have these little workhorses called proteins that can do things like catalyze reactions that otherwise would be too slow to carry out. So these are the machines that make life possible. And proteins are the ones that carry out the bulk of activities required for life. There are some important roles by other biopolymers, such as RNA, and of course DNA plays an important role. But in terms of catalyzing reactions, of making transformations possible, that's carried out largely by proteins. And as we'll see today, we're going to see 3 examples of proteins doing this workhorse stuff. One example will show them synthesizing new DNA, and then we'll show an example where they're turning on and off the switches inside the cell that determines whether the cell goes cancerous or stays normal. And then last, we'll talk about attacking and destroying bacterial invaders. And here's an example of these machines coordinating and at work with each other. This is the little machine that takes the blueprints of the cell, the DNA, and converts it into a message, which then goes out to synthesize more proteins. This is a fantastically complex process. It doesn't involve just one little machine. It involves dozens of machines that are scurrying around and doing all kinds of complex activities. Today, we're going to keep things simple, and I'm going to show you individual proteins. One other thing, proteins' motions are powered by forces. The familiar force, the one that you learned about in high school, was probably something like ATP. ATP is sometimes referred to as the fuel that powers cells. And indeed, it is an important source of energy for some processes in the cell. Unlike, say, gasoline that you put in your tank of your car, when ATP is consumed, it goes through a very different process. It doesn't go through the same sort of reaction. And one of the surprises is that oftentimes this burning of ATP, this hydrolysis of ATP, is not necessarily coupled to the key power stroke of a protein. And let me show you one quick example of this. This is one of your muscle proteins in, say, your bicep. And ATP is being broken apart here to power that muscle. But the power stroke of the muscle is not, in fact, the breaking apart of that bond. So in high school, probably you learned about how this ATP has these 3 high-energy bonds, and each one of them gets broken apart to power things. That's true, but as we start to refine our thinking, we're learning more and more about how that's actually just one step in a more complex cascade. The other thing is that the vast majority of machines inside your cells are not powered by ATP. ATP is kind of the ones that are kind of the special cases. Many of them are powered by other types of bonds being broken. These typically yield a lot less energy. Okay, it'd be like going to your, the gas station, and instead of filling up with high octane gas, maybe you're just putting in the regular. Okay, so a lot less energy is yielded for each one of these bonds that are broken, but it's going to work nonetheless. This is an efficient process. The other force that we don't think about so much is this Brownian motion, the thermal energy that I alluded to on a couple of slides ago. That's going to loom large in a few minutes. Okay? Oh, I should have said this. I didn't. We're now going to get into some more details. We're going to get a little bit more complicated. I want to invite your questions. If you have questions, if you're puzzled by something, don't hesitate to interrupt. And in addition, I'm going to do my best to avoid jargon, and I'm going to avoid complicated descriptions. But sometimes I fail at this. It's a professional hazard. So if I start to lapse and you get confused or lost or something, move your head like this, okay? And then I will know that that's a unique type of movement, and I'll go back and I'll try to describe things a little more carefully, okay? So that's our agreement. So you don't have to feel embarrassed or whatever, just go like this, and I know that I'll circle back and we'll get a longer description. Okay, everyone ready? Okay. The father of this area of using nanometer scale electronics is the great Richard Feynman, winner of a Nobel Prize in physics. And he issued this clarion call to build devices that are at this nanometer scale in 1959 with this classic phrase. He said, there's plenty of room at the bottom. There's plenty of room to build tiny little nanometer scale devices. And today what I'm going to show you is how we're using those types of devices to explore the biological world that I showed you on the previous slides. Okay, so what happens is, as you start building devices that are incredibly small, you get some very unusual properties that are extremely powerful. So for example, these devices have unusually high surface to volume ratios, which grants them very high sensitivity to tiny little perturbations on their surfaces. I'll show you examples of that in a moment. Okay, so this is Richard Feynman playing the bongos. He was also a great bon vivant. And I think anyone in this room would have loved to have met him. OK, so I've told you about the machines. I'm now going to start putting things together. We're going to start putting the enzymes together with the nanotechnology. So what we want to do is I want to build the world's tiniest microphones that will allow us to listen in on those machines at work. OK? And this is totally analogous to, say, listening to a car's engine. Okay, I actually found an example of this. There's something called the mechanic stethoscope, which is used not by my auto mechanic, certainly, but there must be an auto mechanic out there somewhere who can use this to listen to clicking noises and figure out what's going wrong with engines. And in the experiments that I'm going to show you, we're going to use not a stethoscope, but we're going to use something called the carbon nanotube, which is shown in the back of this image. And we're going to use that as the world's tiniest microphone and use this to listen in as proteins dance around on top of the microphone. Okay, so here's the microphone over here. And what we want to do is learn how things work. And then later, we're going to use this to diagnose problems. So the microphones are made out of carbon nanotubes. And all of the experiments that I'm going to show you are done in collaboration with a really talented physicist named Phil Collins. Okay? So Phil's laboratory worked out ways of building microphones based on carbon nanotubes. And what his laboratory figured out was that you can start with, say, a silicon wafer. This is about 4 inches or so in diameter. And then you can use the same photolithography techniques that are used to build things like my cell phone or the laptop. Use those same techniques to place on top of this wafer devices that look like this. And then as you zoom down, you get down to wires that are not connecting. These are called electrodes. They're interdigitated. They don't touch each other. Somewhere out here, we sprinkle an iron molybdenum catalyst that catalyzes growth of these single-walled carbon nanotubes, shown here in this false colored red. Okay, so this is done by Phil's students. And it's done actually in this very building. You can go across the hall and you can see rooms that are called clean rooms. These are rooms that are exceptionally devoid of dust and dandruff and little bits of hair. And the reason is the devices we're making are so small that if little bits of dandruff dropped off of these guys, they might destroy the whole device. The little dandruff particle would be so much larger than the little devices that we're talking about. Okay? So, so this is Phil's laboratory. That's one of the experiments that they're doing. In addition, my laboratory makes the proteins. Okay, so we have this collaboration where my lab makes the proteins, his lab makes the devices, and then we bring them together. To make the proteins, we basically borrow from ancient brewing technology. The same technology that's used to brew beer is more or less what we do on a daily basis. This works really well. You turn bacteria or yeast into little factories that spit out large amounts of your protein, and then we purify this protein. So we use biotechnology to produce the cell's machines in very large quantities. So even though all of the experiments that I'm talking to you about involve only one molecule, it turns out that you can't just isolate and characterize one molecule. It's impossible. That one molecule, disappears, and we don't have the analytical tools necessary to do that. So instead, we make a whole bunch of them, get them about as pure as is capable with state-of-the-art technologies, and then go into the next series of experiments. Okay, so in these next series of experiments, we're then going to attach individual proteins to these carbon nanotubes, which have been turned into sensitive microphones. Okay, and I'm showing you this image because it illustrates that we can attach one protein reliably to these carbon nanotubes. This is an image taken using a technique called electron microscopy, and this protein is attached to a dot of gold, a very tiny little dot of gold, so that we can readily visualize the protein. And you can see we're getting one dot, one dot, one dot, one dot, single dots attached to the microphone with great reliability. And this was exciting to us. Okay, the reason, I'll tell you why it's exciting in a moment. Before I do, I want to remind you of something that probably is obvious, but you never know. That is that we can convert sound into images. And a familiar example is the sound clouds that you may have seen on your Facebook page. Maybe one of your friends wanted to share a song with you or something and gave you one of these sound clouds. But in this takes, say, a musical riff and converts it into a visual that you can sort of reasonably interpret. So for example, this water dance involves a lot of drumming, and you could see each strike of the drum, right? So boom, 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 boom here regularly. And the reason why this is important, the reason why we want to convert the sound into images is that we can get a lot more detail out of those images than we'd ever dream of getting if we were listening to the sound ourselves. Okay, so for example, here I can tell that this drum strike is louder than this one. I can tell that these have a certain regularity. And over here, there's other instruments that are joining in. So there's lots more data. It's a much richer analysis for us if we look at images of the sound rather than listening to the sound itself. This is good news because I have to tell you, proteins are the world's worst musicians and dancers. They have no sense of rhythm whatsoever. We're going to see in a moment that they're completely spastic, completely random in their motions. Okay? So of course, movements make sound. This is familiar. Here is, for example, a dancer. And as she taps her feet on the floor of this dance studio, we can get information about her movements by listening to those foot taps. Right? I can start to get some idea of how it is that she's moving around. Now the thing is, when I just have one dancer on this studio, I can really start to piece together what her movements are like. I can start to figure out how her dancing is working. If, however, I had, say, hundreds of dancers in the studio at the same time, all randomly dancing, which is what proteins would be like, because proteins don't synchronize to music, the sound would be this complete blur of foot tapping. So it's really special that we just have one thing on the microphone at a time. That means we can start to figure out what its dancing moves are like. Okay? In protein terms, this means that we can examine individuals to reveal their unique individuality and also learn stuff that otherwise would be hidden, that would be blurred out by having lots of dancers running around at the same time. In chemistry terms, we say that this teaches us about the dynamics of their dancing. And we can look at things like intermediate motions and the movements that they're actually doing. So for example, I might be able to listen carefully and figure out how far her steps are in this dancing if I had this set up correctly. Okay. In the experiments I'm going to show you, we're going to have, again, this carbon nanotube that is our microphone. Almost immediately, though, we run into a problem. Okay, so stop the presses. This experiment is not going to work. The problem is all of biology takes place in water. I already told you that. And all of electronics hates water. That's why you don't answer your cell phone if you're in the shower. And so our solution there is we cover up all of the electronics with a layer of acrylic nails. It's basically the same polymer that's used in acrylic nails. And that hard plastic polymer encapsulates all electronics and protects it from the water. And then we carve out a little trench in the center of the carbon nanotube using something called an electron beam writer. And so just this center part is actually exposed to the salty water that biology likes to operate in. Everything else is covered up. Okay, so then we attach our individual protein to this. I'm now showing you just that windowed region, and now we're getting really small. Okay, I'm now actually showing you really what an individual protein looks like. So to take these images, we're no longer using electron microscopy. We're using atomic force microscopy, a different technique that lets us get down to really small length scales. Okay, so this little blob in the center of this carbon nanotube, that little blob has the dimensions of the machine, the enzyme that we're going to be studying. We then turn this thing into a microphone and listen in. Okay, so I'm going to show you 3 quick examples of enzymes studied, and then we're going to get into some greater detail in one example that I think is really cool. Okay, the first one is a machine that's required for all forms of life because it synthesizes DNA. Eventually, your cells have to divide or you have to repair DNA. So DNA polymerase is required to do this. This machine has remarkable properties. It's endlessly fascinating. And in this case, we wired into carbon nanotube. Here it is by atomic force microscopy, that little blob right there. And then we listen as this synthesizes DNA, and we can hear each bond being formed. Okay? So here's what our data looks like. So what we're doing, here's the apparatus, here's the enzyme, here's the new strand of DNA coming off of this machine, and each one of these little blips down accompanies formation of another bond as this DNA is polymerized. So it's basically base after base after base. And we can look at things and get, this is usually a bad sign. All right, so we can look at things and get lots of detail about how long it spends in each one of these states. Okay, one more example. All right, this usually means that I have to change out the battery, so I'm going to do that. The next example, though, is a different kind of enzyme. This enzyme I like to think of as sort of an air traffic controller for the cell. This is an enzyme that controls the switches and determines what happens when, and turns on and off cellular processes. And it does this by transferring phosphate groups from one protein to another. This is an enzyme called a kinase. And it, again, plays a key role in cancer. So again, we have the enzyme attached to the nanotube. Okay, so again, we have the enzyme attached to the nanotube. Here's the AFM. That little blob's our protein. Okay? So when we do this, what we see is we see that the enzyme by itself is completely quiet, no dancing. When we then add the ATP that this uses as a source of phosphate, we see these little blips going down. So this is enzyme bound, unbound, bound, unbound, bound, unbound. And you can see that these patterns are pretty random. This thing doesn't spend a lot of time holding on to the ATP. It releases it quickly, et cetera. We then wash away the ATP. We add the peptide that this thing likes to bind, that it likes to, the switch that it's going to turn on or off. And again, we see these blips going down. Okay, it's time for dancing. We now have all 3 things at once. We have machine, we have the ATP that it uses, and we have the peptide, the switch that it's going to turn on or off. And when we have all 3 at once, we could see this thing dancing between 3 different states. State 1, state 2, state 3. So here's 1 second of data. Down here, here's 0.2 of a second. And what's happening is this enzyme is going between an open state with its mouth hanging open, and then it binds either ATP or the peptide, and then it catalyzes the reaction, turning on the switch and turning on the cell's growth process. So in short, this is kind of like a waltz. Okay? So here it is going 1, 2, 3, 1, 2, 3. But then notice that it gets stuck right about there. This is the world's worst dancer. Okay? And I'm glad that I found someone who dances worse than myself, because my wife can attest that it's good that I'm not actually doing this dancing for you. In any case, notice that it's getting stuck over here. It's basically switching gears, and instead of dancing the nice waltz, instead it's switching to like a samba or something like that, and going 1, 2, 1, 2, 1, 2, and trying to decide, go forward or backwards. Okay. So this is really a characteristic of a machine that's imperfect. This is a machine that evolved to be highly regulated by the cell. You don't want the perfect catalyst running around the cell and turning on things randomly. Really, you want something that's very readily controlled and tightly regulated. This indicates that sort of tight regulation because it's not doing the waltz constantly. Sometimes it's sort of hesitating and trying to make up its mind and deciding waltz or samba, not sure. And again, this is characteristic of this type of protein. Okay, so I showed you these two examples to indicate that this approach is general. What I want to do now is switch and look in closer detail at an enzyme that chops apart bacteria and prevents colonization by bacteria of your eyes. Okay, so this enzyme was first described in, again, 1909, which must have been a very special year. The great Alexander Fleming crystallized this protein directly from his own tears. Okay, now that's actually an experiment we should take a moment to reflect on. But the protein is present at such high concentration in tears because it's there to chop apart any bacteria that might land in your eyes. Okay, it's an antibiotic. And of course, Alexander Fleming went on to receive the Nobel Prize for a different antibiotic, penicillin. Okay, so this protein shown here, this is the picture that we use to describe proteins, has a very large domain up here which reaches down to reach this lower domain. This is a big motion. And in fact, actually, the whole protein acts like a Pac-Man that moves backwards and forwards. And we'll look at this in greater detail. In the experiments I'm going to show you, we engineered a special version of this protein. In blue has a sticky foot at its base. And that's where we're going to attach it to the microphone. Okay, so here's what this looks like. This is the enzyme all by its lonesome self. Nothing's happening, no dancing. We add the substrate right here, and boom, it starts turning on. And notice that the current goes up. This is analogous to current flowing through speaker wire. So it gets noisier as it starts dancing. Makes sense. And in here, we can get lots and lots of detail about the motions that the enzyme is doing. Okay, so if we zoom in on this, we're going to learn all kinds of things. Okay, so before I get too far into this, I want to show you some controls. So remember, we're not actually visually seeing this. Instead, we're inferring the motions by listening to the noise. So control experiments are crucial to prove that it's doing what I'm telling you it's doing. Okay, so let me show you a couple of controls, and we have lots more that are backing this. In this control over here, this is the food that the enzyme likes to chew on. So this enzyme, lysozyme, chews on the cell walls of bacteria, and we isolate little bits of those cell walls and feed it to the enzyme. Those cell walls by themselves, in the absence of enzyme, nothing. Nothing. There's no dancing around on the surface by these guys. Enzyme by itself, again, nothing. This is an overlay of the control, so you can really see it's nothing. When we add the cell wall fragments together with the enzyme, there are these pulses going up in the current. And we interpret this as enzyme open, closed, open, closed, open, closed, open. Sometimes it stays open for long, sometimes it closes for a short amount of times, other times for longer times. This is truly a random dancer. Okay? Now, one other thing about this enzyme, I should tell you that we compared our results with other experimental results, and they match in terms of timing and variability, et cetera. The other thing we noticed, and this was something that had already been known, is that this enzyme has 2 speeds. It's sort of like a 2-speed car. And there is a very rapid opening and closing state, and then there is a much slower opening and closing states. These are roughly the same amount of time. This is a third of a second. This is a quarter of a second. But you can see there's 10 times as many opening and closings under this rapid switching than there is normally. Turns out that this sort of rapid motion is the enzyme gnashing its teeth and not actually catalyzing any reactions. It's randomly opening and closing and not actually doing anything. Okay, it's just random, non-productive movement. And I'll tell you what that random non-productive movement is in a moment. This is the motion that the enzyme uses when it's actually chewing and making progress going across the surface of the bacteria. Okay, so I said this before, but our experiments are compared then with other experiments. And in short, everything that was known previously we saw as well, but we also learned new things. Okay, and this is the fun part of being a scientist. This is what I'm going to share with you today. Okay, before I get to the new stuff though, I want to tell you about a couple of things that we saw. We've already published these, so they're no longer new, but at the time this was a breakthrough for us. One thing that we saw is that this enzyme grabs on to the little cell wall fragments and does not let go. It takes bite after bite after bite. It's sort of like a bulldog. It grabs on and then just keeps going across the top of the cell. Okay? So it has these two states, again, the rapid chatter state and then the processing state where it's actually chewing. When we compared two different foods, so we fed the enzyme two possible foods. Either a linear food over here that was chemically synthesized by Larry Blazek and coworkers, or a cross-linked food. This is actually what the cell walls actually look like. We saw a big difference in that non-productive state. Okay, so in short, you can see that this guy is spending a lot more time kind of gnashing its teeth. So the gnashing its teeth is a strategy that the enzyme uses to get past the cross-links that hold together the cell wall. The cell wall is sort of like a net. It looks like this, and that net is held together by crosslinks. And this gnashing non-productive state is going to be interesting to us in a moment. Okay, everyone still with me so far? Okay. So back in January, so at the beginning of this year, the laboratory made a really important and significant step forward. This was done in Phil's laboratory by a graduate student named Max Akhtarov. And I apologize, I'm not doing justice to all of the work that Max did. But these next experiments required literally years of effort to set us up. Okay? And it involves stuff like working out all of the electronics and avoiding parasitic capacitance and things like that. But long story short, what Max did was he installed much faster amplifiers in our experimental apparatus. By the way, this is what the experiment actually looks like when it's on the lab bench. Okay, so over here we have these lines that go in for fluid delivery. This is where the little cell wall fragments get fed in. We have little pumps over here that are feeding all of this. And then over here in the very center, there's the little chip that I showed you earlier. In the very center of that, there would be the single molecule, the single enzyme attached to its microphone. Okay, but the important thing that Max did is that he sped up the resolution, the time resolution of our experiments into a regime that's otherwise not explored. Okay, so we're down here in this single-digit microsecond regime of protein motions, which is really exciting. This meant that for the first time, we can start watching enzymes open and close. Okay, and I should tell you, single molecule protein experiments have not been able to see that previously. So we started looking at opening and closing. Here's 3 examples of closing events. Here's 3 examples of the enzyme going between a state of closed and then to open. And what we saw was a vast diversity. So in the same way that the enzyme is a random dancer, speeding up, slowing down, it's also a random mover in terms of how fast it opens and closes. Furthermore, it doesn't seem to have a memory. If it closes quickly, it will open maybe fast, maybe quickly, but it doesn't seem to care. There's no memory of if it starts running at one speed, it stays in that speed for a long time. No, this thing is totally random. The other thing that we noticed is that 10% of the time, there was this intermediate state between open and closed. And we saw this both for closing and for opening, roughly 10% of the time. Now, this is when things started to get weird. And this is what I want to share with you today, because this really took me a summer to figure out. We found a lot of coincidences when we started looking closely at our data. I asked Max, what is the speed of opening and what is the speed of closing? And he looked at me and said, it's 37. And I said, oh, that's fine. 37 for opening or 37 for closing? Both. They're both roughly 37 microseconds. It takes about 37 microseconds to close, 37 microseconds to open. That seems very weird, right? Because if I'm eating an apple, it's going to take me a little longer to bite into the apple than it is to, just open and close my mouth. Okay? And we saw this 37 microseconds for both catalytic states, where it's actually doing chewing, and also for non-productive states. Furthermore, we saw essentially identical distributions of opening and closing times. That's a histogram shown down here. You can see these lines are overlapping each other. This is actually for closing states, and I'm comparing catalysis and non-productive. Again, these two are the same. Also very suspicious. And then finally, you remember I said there were 10% intermediates? Well, we see that regardless of if the enzyme is eating or just gnashing its teeth. Both times there is this weird coincidence. And that's just too many coincidences to possibly not have an explanation. In the words of the great Sherlock Holmes. So here is Sherlock talking to his brother Mycroft Holmes. Oh, Sherlock, what do we say about coincidence? Sherlock says, the universe is rarely so lazy. Right? And then this is better stated by one of my favorite actresses. I don't believe in coincidences, says Juliette Binoche. She's an expert. She's right. This has to have some explanation. Okay? So we started looking at this in greater detail. And what we found is that lysozyme harnesses Brownian motion, that little thermal energy that I talked to you, as a way of generating its opening and closing. That's why they're the same. It doesn't use the energy for stickiness or something like that to open and close. Instead, it harnesses exactly the same amounts of energy for both opening and closing from Brownian motion. On the other hand, bond breakage powers sliding along the polysaccharide. So here's the enzyme Pac-Man open. It breaks the bond as it closes. This bond is now broken. This then has a 1 kcal, sorry, I'm getting ahead of myself. This then slides along this polysaccharide to the next bond and then pops open. Now, here's the thing. I'm showing these arrows because actually the surface of the bacteria that's chewing apart has directionality, and this enzyme can only slide in one direction. It also opens and closes along a highly restricted path, hence the 10% intermediates that are formed. Okay, now I can't resist. I have one more chemistry slide. This one is a real chemistry slide, and I'm going to tell you essentially the same thing that I just said on this general audience slide, but now I'm going to tell you in the chemistry detail. Okay, so brace yourself for a little bit of jargon. In short, this illustrates the principle of microscopic reversibility. What we're seeing is that the enzyme has a certain amount of energy required for closing, and then it closes on one glycosidic bond, and then it opens on an essentially identical glycosidic bond, so that both processes are energetically equivalent to each other. It's basically doing the reverse reaction when it reopens. Okay? So we can measure exactly how much energy is released by this hydrolysis. It's about 1 kcal per mole, which is a minor amount of energy. That minor amount of energy, about the strength of a hydrogen bond, is enough to allow the enzyme to slide along the polysaccharide and reach the next identical glycosidic bond that it's then going to hydrolyze. Okay? All of the constraints, all those coincidences that I showed on the previous slide provide evidence for constrained motions, for a ratcheting type effect, and thus we conclude that lysozyme is a bridge-burning Brownian ratchet. Okay, this is a special class of enzymes that usually is used for motor proteins and things like that. This is a very rare case of it actually being applied to chewing apart a biopolymer. To my knowledge, this is only the second example. Okay, so I want to conclude, and then I'll thank people who made this work possible. I've been telling you about listening to normal proteins to try to understand how they work. But we could just as readily listen to the abnormal ones and learn stuff about human disease and try to address human frailties. For example, we want to understand what happens in cancer-associated mutations and compare those against normal proteins for something like protein kinase A, the air traffic controller that I showed earlier today. This is an ongoing experiment going on in the laboratory right now. In addition, proteins also solve problems for humans. They're used routinely. It's something like a $50 billion a year industry for producing proteins. And this gives us a new opportunity to start to tinker with these proteins and understand at the level of atoms and bonds how they're working. And so we want to develop better proteins using an iterative process of listening to them, tinker with them, listen again, et cetera. So in the future, we're moving towards these types of experiments over here, but we're also going to be moving towards more complicated processes. Earlier, I showed you, for example, the much more complex process when DNA is transcribed into RNA, and that involved large numbers of components. We're setting up for those sort of multi-component processes right now, and I'm very excited about running those types of experiments involving, for example, inhibitors, mutations, steric crowding, et cetera. So I think the future is going to be really neat. All right, I need to thank important people who made all this work possible. Foremost, I'm very, very fortunate to have started a collaboration with Phil Collins greater than 10 years ago. We've been collaborating ever since. He's an amazingly rigorous and deep-thinking physicist, and it's been my good fortune to collaborate with him all these years. His laboratory is shown here. This is actually Phil over here in the center in red. The people who did the experiments that I talked to you about today were Max Oktarov, shown here. He did all of those high-speed measurements that I showed you. The protein kinase A, the air traffic controller protein, was done by Patrick Sims. And then Yanqi Choi played a seminal role in kicking off this project several years ago when he started figuring out how to get proteins reliably attached to the nanotube and take measurements from them. He did a lot of the work with DNA polymerase that I showed, and also a lot of the lysozyme experiments, essentially all of lysozyme except for the ones that I credited to Max. In my laboratory, I'm really fortunate to work with a very talented team of truly outstanding students. They make me run to work every day, and I love working with them. So I particularly want to thank the team that worked on the experiments that I showed you today. Miriam Iftikhar works with the air traffic controller kinase. Caitlin Puglisi works on DNA polymerase, a protein that I mentioned, the one that synthesizes DNA. These experiments are also joined by research scientist Dr. Sudipta Majumdar, who just joined the laboratory. Former members of the lab who made this work possible include John Coronius, Isa Moody, Tivoli Olson, who laid the groundwork for what I showed and were in many of the publications that I alluded to. In addition, I want to thank all of you, the taxpayers who made this work possible. I very truly work for you, and I'm really grateful for that support. It makes this work possible, and without it, none of this would have been possible. So it's a real privilege for me to tell you what I'm doing with your money. I'm particularly grateful to the National Science Foundation, which started the earliest funding of this work. This was then followed by support by the National Cancer Institute, and then more recently, the National Institute of General Medical Sciences. So thank you very much to everyone. I really appreciate this opportunity to talk to you. Thanks.
Any questions?
Yes.
In your description, you're talking about using nanowire to listen.
Right.
Things. What happens if you actually listen, making an audio track?
It's worse than Schopenhauer. I mean, it's really, there's no rhythm, there's nothing. And it's worse, it's even hitting just one note or one very close other note. It's just like random bonging by a chimpanzee or something. It's the worst. So I've actually tried it. It's just not, it's the least interesting sound I've ever heard. So, yeah, Ken.
So following up on that, you use noise ambiguously a little.
I'm really glad
so
you asked.
there's good noise and bad noise.
Yes.
How do you distinguish the two?
Okay, so Ken's question is a perceptive one. The carbon nanotubes are very, very sensitive to tiny little perturbations in their environment. And we're fortunate that the signal of our protein moving is a larger signal-to-noise ratio than just those little tiny things that are constantly happening with carbon nanotubes as they respond to small little changes in the environment. In addition, you didn't ask me this, but I know everyone must be thinking about this, which is, do proteins really make noise, right? Because all of you, are here, and you don't have this sort of constant, din in your ears, right? Okay, so actually what we're listening to is we're listening to charged residues on the surface of the protein that are in motion. Those charged functionalities make noise. And an example of this would be a flame. If I had a little flame, a candle, say, over here, and a gust of wind came in and the flame moves around, you'd expect it to make a guttering noise. That guttering noise is caused by the plasma ions in the flame moving around and making noise. And so we're listening to those charged residues that are analogous to the plasma ions that are moving around close to the carbon nanotube. So it actually is a pretty good analogy. So I answered questions. Did I answer your question? But I also talked about something else. So, okay. Yeah.
For the DNA polymerase.
Yes.
Does it make a different noise or dance differently depending on what base is being added?
So you really want to know, can we do DNA sequencing?
Yeah.
And yeah, that would be the coolest thing ever. The short answer is there are some small differences. But there's so much variation at the single molecule level that the variation tends to overlap with each other. So we can't sufficiently discriminate amongst the bases. It's possible, we have some ideas to change that, but for now, the answer is no. So thanks for asking. Yeah.
You mentioned the 37 microseconds.
Yes.
Knowing what you know now, could you derive that from first principles?
I could if I talked to one of my distinguished colleagues over here who could model something like that, which would be really interesting. So thanks for asking. And thought I saw a question back there. Oh, the question was, could you derive that 37 microseconds from first principle? And I think the answer is yes. I couldn't do it, but I think one of my colleagues could. So, yeah.
Since the motions are so random, how does the protein have such good control?
Okay, so Ev's question is a really good one. Ev is asking, this thing is in this 15,000 mile an hour hurricane, the equivalent of a hurricane, coming in all directions. How does it actually harness those into just one set of motions? This is really the beauty of evolution. The enzyme evolved to harness Brownian motion. It evolved a joint, a hinge really, that only allows motion in that direction. And the motion is in a very constrained pathway. So all of the motion requires a whole set of hydrogen bonds, and everything, all the side chains have to be lined up in order for the enzyme to open and close. So it's really a remarkable thing. Yeah, Bill?
Have you thought about using this in a non-biological system?
We have.
It'd be a little simpler.
Yes. Yeah, so I have a postdoc arriving.
An olefin polymerization. Could you watch that and get information about it, but also information about your technique?
Yeah, okay, so Bill's question is, could you use this for a non-protein catalyst? And I have a postdoc arriving in February whose plan is to do exactly that, and I can't wait to do it. So great question. Jenny? Oh, I love that question. Okay, so Jenny's question is, could you, say, put AC current in the nanotube to drive the protein to go faster and slower, like a marionette, where you're actually controlling it? So we've tried that, and so far it hasn't quite worked, but we're not giving up yet. That would be really cool. So, and I think it's reasonable. So, Sam?
Because you were saying that the protein has a very specific motion.
Yeah.
It does this chemistry. So if you were to attach the protein in a different part of the, that blue sticky stuff that you
Yes.
if you put that in a different spot, would it affect how it does the chemistry?
Absolutely. So Sam's question is really good. What he's asking is, does it matter where you attach to the enzyme? And the answer is yes. When we so we typically start our experiments by designing and synthesizing 5 different attachment sites. And we look for ones that have relatively simple data interpretation. Because sometimes we attach to other spots on the protein that have additional motions, say loops that are flipping around at a faster rate than simply an enzyme opening and closing. And when we see those really complicated motions, it's very hard for us to deconvolute the data. So we simply move on to a different attachment site. But it's a great question. It makes sense. Yeah.
Do you believe you can apply a particular resonant frequency to induce a certain conformation in the protein?
Okay, so the question is a little bit like Jenny Yang's question. Can we drive the protein into specific conformations that are perhaps not biologically relevant, or get it to dance to our own tunes and not let it do random dancing? I think we could, but we haven't gotten it to work yet, so it's possible. We're just not there yet. So. All right. Well, I really want to thank everyone again. I'll hand things back to.
So, pretty fabulous.